Hello and good morning. I hope you all are doing well. Thank you to all of you who sent me an email to let me know that you're doing well. I really appreciate it. It's good to hear from all of you. So if you have some time, please send me a message that everybody is fine. Your loved ones are well. I'm doing fine. So far, our labs are completely closed now, so it's all work from home. Okay. So let's start with done with transcription per se, because we talked about both prokaryotic and eukaryotic initiation, elongation, and termination. Now, today's topic mostly covers the RNA processing. So we started talking about RNA processing along with transcription in eukaryotes, because in eukaryotic transcription, the cap on 5' is synthesized as soon as RNA comes out of the RNA polymerase. And then poly A tail is also added before the RNA transcript is complete. The polyadenylation takes place by use of a specialized polymerase that will add those poly A or adenine towards the tail, about 200 to 250 adenines at the end of mRNA. We mostly talked about mRNA because we're talking about gene expression here in this class, so that's what we mostly will be focusing on. So today's topic covers RNA processing, so mostly splicing. I will talk about that in a minute, and this is most of this portion is from chapter 13 and chapter 14. All right. Okay. We didn't really cover a whole lot of RNA, the functions of RNA pol 1 and pol 3 that make ribosomal RNA or transfer RNA. We covered very briefly. So if you want to learn more about these processes, this is a very good paper here, this Transcription by RNA Polymerases 1 and 3. It summarizes a lot of literature because it's a review paper. So if you are more interested in these topics, I would suggest you read this paper. Okay, so as I mentioned, we will be talking about splicing today. What is splicing is, let's look at the gene structure. This is the gene structure. This is the genomic DNA. So we have the promoter region. That's where the RNA polymerase will come and bind along with multiple other transcription factors or other proteins, along with all those the transcriptosome that will be formed here. In addition, so that's the promoter region. There, it will start copying this sequence on the template DNA. The template DNA has these sequences before the first exon. So sometimes this is the first intron. Most of the time, it's an untranslated region. It's called 5' UTR, 5' untranslated region. Then you have your multiple exons depending on your gene, and then you have introns between those exons. Most eukaryotic genes have at least one intron. So in this example, you have 3 introns. You have 1, 2, 3, 4 exons, and then 3' untranslated region or 3' UTR. When during transcription, first in the pre-mRNA, this sequence will be copied as such. So this is 5' leader or 5' UTR, your first exon, intron, exon, intron, exon, intron, exon, and this is your non-coding region at 3' end or 3' UTR. Before this pre-mRNA becomes a mature mRNA or spliced mRNA, these introns needs to be taken out and these exons need to be joined, because these exons are the portion of the genome, portion of the gene that will code for the protein sequence. And this need to be accurate, because if it's not accurate, then it can change the coding sequence. That will change the amino acid sequence of the protein. So there are, as I mentioned earlier, there are multiple RNA processing events in eukaryotic RNA. This includes splicing, that we will talk today, capping that we talked on Wednesday, and polyadenylation also talked on Wednesday, where the A's or adenines are added towards the 3' end of the mRNA. All of these steps happen in the nucleus. So this before the mRNA is transported into cytoplasm. For translation to happen, all this need to occur within the nucleus. This hnRNA or heterogeneous RNA, that's heterogeneous RNA is the RNA that's include introns. This will associate with different proteins almost as soon as it emerges from RNA pol 2, and it remains associated with these proteins as long as it exists. So we talked about this capping enzyme. This capping enzymes or these proteins, they bind to this C-terminal domain of RNA pol 2. The C-terminal domain or this tail is very important in RNA pol 2. The phosphorylation at different sites will determine the capping and promoter escape. So, for example, here the phosphorylation of serine 5 results in capping. Dephosphorylation of this site will mean promoter escape. Okay. So the capping enzymes come and bind to it. This determine the capping, the formation of 5' cap in mRNA. Complements of splicing machinery that we will talk today also come and bind to this tail. And polyadenylation and cleavage factors, we talked about those as well. These polyadenylation and cleavage factor, they come and bind to the sequence here on the gene. Once the mRNA moves to that polyadenylation sequences, these proteins will come and bind to mRNA, and that's where it will dissociate and make the poly A tail. These modification proteins are delivered to this heterogeneous RNA by the CTD of RNA pol 2. That's what this CTD is very important. So this chart here, this graph here shows number of introns per genes on an average, and these vary among eukaryotic species. So here you can see that in Homo sapiens or the model organism, mouse and Arabidopsis, there are about 6 to 7 introns per gene, but that's an average. There are some genes that have hundreds of introns, and there are some genes that have only maybe 1 or 2 introns. So these averages. This is on an average. Most genes in eukaryotes have introns. Some organisms have fewer introns. For example, yeast here has less than 1 intron on an average per gene. So again, some genes will have introns, some genes might not have introns, or some genes might have 2 or 3, and some have just 1. So you can see that similarly this varies from organism to organism. C. elegans have around 5. Drosophila melanogaster has around 3. Plasmodium falciparum, plasmodium, these are protozoan, single cell eukaryotic pathogens that cause malaria. They have about 1 intron per gene. So all these, just in short, most eukaryotic organisms have introns in their genes, and those introns need to be removed before the mature mRNA is formed and transported to the cytoplasm. How these introns are spliced? These introns are spliced by formation of a spliceosome, because introns must be removed before the transcript can be translated into protein, and this process of intron removal is called RNA splicing. RNA splicing need to be precise to keep the reading frame as such to make the correct protein. These introns are removed by 2-step process that involves this formation of lariat here, and there is this free hydroxyl group on the 5' end of intron or 3' end of exon that will make a break at the other end of the intron to remove this intron completely. We will talk about this in detail in next slides. So here this is an intron lariat is formed. This intron lariat stays with the proteins. It gets degraded. Exons will then be joined together. So these exons will be joined together. This lariat formation happens at this branch site. This branch site is most always an adenine. So this is A here. This is called a branch site. Okay, so we'll talk about this in the next slide in more detail. So here this shows in a little more detail what's going on at these splice sites or intron-exon boundaries. Intron-exon boundaries, we can predict intron-exon boundaries by different softwares, by using different algorithms, because most always, so there's a consensus sequence for exon-intron boundaries. At most intron-exon boundaries, there will be a GG. This is a common feature, and that's how we know that this, when you have a gene sequence, when you're predicting how many introns, how many exons are there in this gene sequence, you will look at these consensus sequences for the boundaries of introns, exons. So here this G here at the 5' end of intron is quite important, because this G binds with this branch site A to make this lariat. Okay. These are the consensus sequences. You don't have to remember the consensus sequences as such, but you should know that there is that this is usually there G at the 3' end of the exon, first exon, and a G at the 5' end of the intron. Okay. So so far we just talked about a general mechanism, the two-step formation. In the first step, there is a break on between intron and exon boundaries that forms a lariat at the branch site, and then the 3' hydroxyl group of the first exon will make a cut at the 5 to 3', 3 to 5, between the 3' of the intron and 5' of the next exon to remove the intron from there. But now we're going to talk into more detail how exactly that happens. This process of splicing, this splicing from here, this intron from here to here, it requires consensus sequences on this heterogeneous RNA. This is the consensus sequence here. So AG-GU, AG part of the 3' of exon, and GU is part of the 5' of intron, of the first exon intron. And then again, this is the same sequence here at the boundaries. This is AG, and this will be GG. In addition to these consensus sequences, there are 5 small nuclear RNAs that play a role in splicing. These small nuclear RNAs are called snRNAs. These snRNAs are U1, U2, U4, U5, and U6. In addition to these 5 snRNAs, there are small ribonuclear proteins, or SNRPs. So these are the proteins that binds to these small nuclear RNAs. And then there are additional splicing factors that assist in this process. These splicing factors, or these SNRPs, these are loaded by RNA pol 2, by RNA pol 2 that carries these on its CTD. So again, the CTD is really important. The spliceosome complex consists of is RNA and protein, and it carries out splicing. It includes snRNAs, SNRPs, and additional about 100 proteins. This consensus sequence on exon 1 is usually AG, GU. That's this hash here means exon intron boundary. So this is part of the exon, this is part of intron. This is that middle sequence of the intron. A is the branch site, and then this is the 3' end of intron and 5' end of exon. So these GG makes these exon binaries. So again, here is it shown. So how does this how these snRNAs and SNRPs and these associated factors, these all come together to remove the introns, is what we're going to talk about now. The SNRPs bind to these defined sequences at 5' and 3' splice junctions. See here. So these are those your snRNAs, U1, U2, U4, U5, and U6. So U1, so these U1 finds this intron exon boundaries. This binds here. This is SNRPs, bring them here, of course. This binding is mediated by RNA in the SNRP that is complementary to the consensus sequence of heterogeneous RNA. So remember, those SNRPs are RNA protein. These are those. That's what. Let's see. That's what a SNRP is. This is a small ribonuclear protein. So they have. These are proteins that have RNA sequence associated with them. So these RNA sequences are complementary to the consensus sequence on this heterogeneous RNA or hnRNA. These find this consensus sequence and bind to the consensus sequence. The catalysis will not happen until the whole spliceosome is formed. So this is just the beginning of the spliceosome. This is assembling here. So in the first step, the U1, so U1, U4, and this auxiliary UTAF, AF means auxiliary factor, and the BBP, this branch binding protein. These all are required for assembly. What these do is they help stabilize and regulate the activities of these SNRPs, because SNRPs have the catalytic activity, so they have enzymatic activity for splicing, and those will cut those, make the cuts where these cuts need to be made, especially here at this end. So U1 binds to it's U1, the SNRP contains this U1 that will have the consensus sequence, complementary sequence. It will bind there. It will the U1 will bring the U1 there. U1 binds there. Branch binding protein will bind to the branch. That's the branch site is where this A is. This auxiliary factor 65 and 35, they bind towards the 3' end. So these auxiliary factors are mostly just to hold the place until the other snRNPs or snRNAs come and bind here. U2 comes in. Once these all bind, U2 comes in, U2 will bind where the BBP or branch binding protein was bound. So BBP will be dissociated, U2 will come and bind here. Binding of this U2 to the branch site changes the structure of DNA, or moves makes this kink, provides this kink in the DNA that it will do is this A is now outside. It just comes out of the double helix. So it makes this little bubble here. Once U2 is bound, once A is present for a lariat formation, this so this U2 presents this A for lariat formation here. Once U2 is bound, A is present for lariat formation, then this assembly of these other SNRPs, these U4, U5, and U6, these will come and bind. At this point, this auxiliary factors aren't required, so these auxiliary factors will be removed, and U4, U5, and U6 will bind here. These SNRPs together, they provide structural and enzymatic activity for splicing. So this image is not completely correct, and I will just talk about that in a minute. Why? So here, as you can see, this U5 hydrogen bonds with both 3' end of exon 1 and 5' end of exon 2, but in this image it doesn't show all the way here, so it still shows the place where the auxiliary factor was, but since the auxiliary factor is removed, the U5 protein covers this whole position here. So just keep in mind this U5 binds to here as well as here. So you can see here it's bound to both through U6 is bound here, but it also binds here. U6 is is very important as well, and I will. You can see it here. Once this cut is made at the between this intron exon boundary, this exon is kept. It's still associated with the complex through binding to U6. This 5' end of intron will go and bind to this branch site A that already had this, that was presented by U2, and this will bind here. This G will bind here. Now this position here has a free hydroxyl group. This free hydroxyl group will then get dissociated from U6, will make a cut here, and this intron will come out. This intron will still stay associated with U2, U5, and U6 NERPs. Exons will bind together to make one exon instead of two. Okay. So once this exon, this intron is removed from here, these dead box RNA helicases, these use ATP hydrolysis to disassemble the spliceosome complex. Intron is degraded because intron is not needed anymore. The SNRPs are recycled for further splicing, of next mRNA, and then mRNA is this mRNA here is transported to the cytoplasm. This mature mRNA. These are here. It only shows 2 exon, but it is the complete mRNA with the 5' cap as well as the poly A tail. Just wanted to make sure here that we all are on the same page. So these dead box RNA helicases, these are a class of RNA helicases. These proteins are called dead box proteins because these were found to play a role in RNA processing. This dead box means the one of the protein domain has to have this box. That means these amino acid sequence. So D will be your aspartic acid, E will be your glutamic acid, A is alanine, and then the aspartic acid again. So that makes it a DEAD box. So these RNA helicases are very important for RNA processing, for splices, for this splicing of introns. Okay, so here this is the whole mechanism. Just once again, these SNRPs U1, U2, U4, U5, U6 play an important role in splicing of intron from exon. U1 comes and binds to the intron exon boundary. It has the consensus sequence to. It has a complementary sequence to the consensus sequence on these boundaries. That complementary sequence will bind here. Once this U1 is bound here, the branch binding protein will come and bind to A, and then auxiliary factors U2AF6535 will also bind towards the 3' end of the intron that need to be removed. These BBP and auxiliary factors will dissociate, and instead of this other SNRPs U2 and U6, U4, U5 will come and bind. U5 will bind to both 5' intron here and 3' end here to cover the whole intron. Binding of this U2 will change the conformation of DNA, and the change in conformation of DNA will lead to this A sticking out of the helix. This A will be presented to form a lariat once the splicing happens. So here, once all this spliceosome is associated, U1 dissociate because U1 isn't needed anymore. Instead, this U6 will has enzymatic activity. It will make this cut here between intron exon boundary. This free end of exon 3' exon now will stay attached to U6. The free end 5' end of intron will come and bind to this branch A here to make this lariat. This 3' end the free hydroxyl group at 3' end of exon will make a cut here at the intron exon boundary here. This will now take the intron out. Exons will join together to make the mature mRNA. these SNRPs will stay associated with intron. The dead box helicases will come and bind, and they will degrade this intron, and these SNRPs will be reused or recycled for the next mRNA. There are sequences in the exons that are important for the splicing to happen. These are called exonic splicing enhancers. These sequences reside within exons. These bind to serine arginine rich proteins, or SR proteins. These SR proteins are example of splicing factors, and these can recruit U1 and those auxiliary factor U2AF 6535 to boundaries as well. Okay, here shows these are the sequences in exon. These are called exon splicing enhancers, or ESEs. These sequences they bind specifically to these serine arginine rich proteins. These serine arginine rich proteins act as splicing factors, and these can bring these auxiliary factors and these NERPs to these sections, to these boundaries. Okay, let's see. So here, this shows this is an intron, this is exon, this is intron, this is exon, intron. So one gene can have multiple exon introns, but it's not always spliced as first exon, second exon, third exon, fourth exon. The splicing is happening simultaneously, and it can also be that your intron third spliced out first, and 1 and 2 are spliced out later. So there is no tracking mechanism. They all are going to be spliced out. It doesn't have to be in a consecutive order. Okay. Alternative splicing is another very important topic, very important mechanism that can bring diversity to gene functions. So now we have, as I mentioned, we have only about 2% of our genome codes for proteins. So we have about 20,000 genes and that will code for protein. Although our genome is huge, but out of that, only a fraction of that genome is coding for protein. However, these 20,000 genes can make hundreds of thousands of proteins. And how do they do that is because most of eukaryotic genes can be alternatively spliced. And that alternative splicing means you have the same gene, but it can make different protein sequences. Protein sequence can be shorter. You can have one protein might be coded by all 5 exons, one is coded by 4 instead of 5. So that's alternative splicing. So it's the selective use of splicing sites to yield different mRNAs from the same heterogeneous RNA is called alternative splicing. It results in exon skipping or sometimes alteration of exon structures. So here are these different examples of alternative splicing. The alternative splicing requires specialized splicing factors. So these are members of these heterogeneous nuclear ribonuclear proteins, or HNRNPs protein families. These bind to exonic splicing enhancer that we talked about in the previous slides. The sequences, those are especially rich in, they especially bind to these serine and arginine rich proteins. These usually bind to these exonic splicing enhancer or exonic splicing silencers. So if there's a silencer, that means there won't be any, if the protein binds, if these ribonuclear proteins bind to these splicing silencers, this will avoid splicing of that portion of the gene. These HNRNPs are typically specific to a cell type, so different cell types make different splicing isoforms of the same genes. So this is an example of a number of different ways splicing can happen. This is our DNA template sequence. We have here 3 exons and 2 introns. The transcription happened transcript, and this is primary transcript of mRNA that has all the introns and exons in it. And now in normal mRNA, what will happen is you will take these 2 introns out, and these 3 exons will be together to make this normal transcript. So here these 2 are spliced out, these 3 together exons, it's a normal transcript. What if you skip an exon? So if splicing happened here and splicing happened here, but it doesn't happen here, so that will lead to 1 and 3. So there is no exon 2. So this will be a smaller mRNA, so smaller protein. It will be a different protein as well because it's lacking that middle sequence. So it's this protein will be similar to your normal protein at the 5' end and the 3' end, but not in the center. This is exon skipping. Exon extended. In this case, instead of removing this intron from exon-intron boundary, you're only removing a portion of intron, and portion of intron it's still associated with the spliced mRNA, with the protein, with the mature mRNA. So that will increase the protein size from the normal protein, and this is an extra. These are extra sequences. This is called exon extended. Another way of splicing is intron retained, where one of the intron is capped, and this intron will now be translated. Alternative exons, where you have either 1 and 2, no 3, or you have 1 and 3, no 2. So these can be 2 different splicing variants of the same gene. Okay. So this alternative splicing leads to different protein forms. These are called isoforms of the same genes. So these proteins are fairly similar, but not exactly similar, because they either lack a portion or they have an additional sequence of additional amino acid because of those additional introns or portion of the intron. So this is here's a good example of alternative splicing. This is an example of a eukaryotic virus. This is in fact monkeys, SV40, synvis virus 40. This TAG or small lowercase TAG protein. So this is these proteins are translated from alternatively spliced form of the same heterogeneous RNA. The normal or canonical splicing yields an mRNA that will lead to this TAG form. So this is the normal protein, normal mRNA splicing. But this TAG causes cell cycle progression and transformation, usually. So here, if we look at this, this is your DNA sequence that will be copied in mRNA. This is an exon 1. This is exon 2. There's a stop codon right here after exon 1. So in the normal splicing, this intron will be taken out. So this will be exon 1 and exon 2. Right? So this is your normal. This is exon 1. This is exon 2. The stop codon is intron, so it doesn't matter. This will be spliced out anyway. That makes your TAG the normal protein for cell cycle progression and transformation. However, there are some of these sequences here that you can see. So this in this case, that your intron exon boundary has shifted towards the 3' end of the intron. So this is the normal boundary. That's where the intron exon will be spliced. This is a splice boundary for here. But in this case, instead of here, the splice boundary has shifted here. So now you have splicing here and splicing here and splicing here. So only this portion of the intron is removed. What would you assume is this will be a longer protein, but because there is a stop codon right here, this will be a shorter protein. And so let's see here. So we have this one exon and then this exon here, but when this in the mRNA, but since there's a stop codon here, the protein will only be formed from here to here, and it will stop here. So this is a smaller protein. This is a truncated protein of the same sequence. So so if this TAG prevents apoptosis response, so this will lead to more cell progression. Another example of alternative splicing is DSX or double sex pre-mRNA in flies. So here, this is male, this is female. This TRA2 is a transformer protein. This is TRA2. The gene is involved in regulation of sex determination in many insects. So what happens is this here, when this TRA binds to this exon. So we're not talking about exactly about TRA protein. We're talking about this double sex protein. So this is double sex, has a couple of exons. So this is exon 3, exon 4, and exon 5 in males. Similarly, it also has exon 3, 4, 5 in female. But what happens is this gene is expressed in both male and female flies, but because of alternative splicing, the protein they have different isoform of proteins. The isoform DSX female in females is longer than the males, and that's because this is longer because here you will see that it's longer in males and it's shorter in females, and that's because here in males they do not so this exon 4 is deleted. It only goes from exon 3 to 5, whereas in females it's opposite. It's exon 3 and then exon 4, so this exon is removed. But this because this TRA binds here, and this TRA does not allow this to be spliced, so this exon is not the part of the female DSX protein. So the male will have both exon 3 and 5 and it makes this larger protein. Females will have only exon 3 and exon 4 and makes this shorter protein. So that's the female version, and it's important because this determines the sex in the flies. Okay, so now let's talk a little bit about RNA transport. So RNA needs to go out of the nucleus into the cytoplasm in order to be translated, because translation happens on the ribosomes in cytoplasm, right? So RNA transport for transport of mRNA, this mature mRNA, needs to be exported out of nucleus into the cytoplasm where it will be translated. So this is mature mRNA now. This is a 5' cap. It has a poly A tail, and this is all exons all together. These are the binding proteins that bound to the mRNA. So this process is this transport of mRNA is selective for mature mRNAs because introns aren't going to be part of the protein, so they don't need to be transported. And since introns makes the large part, the introns can be huge, can be really large, from 100 base pair to a couple of thousand base pair, sometimes even 80,000 base pair intron within a gene sequence that by itself, just the exons aren't going to be that large. So we don't have to transport all that large sequences because those are not going to be translated. So these intronic RNAs, these will be retained in the nucleus, and these will be retained with these HNRNPs, or if these were spliced differently, or with just the SNRPs. So these introns will be kept in the SNRPs, and these will be broken down by the DAAD box helicases, whereas the mRNA will be transported out. This mRNA is now associated with multiple different proteins. These proteins include those SR proteins, those serine arginine-rich proteins. These SR protein are supposed to provide an address label. These are supposed to tell the mRNA where to go. It takes them to the ribosomes for translation. These proteins that are bound to this mRNA, these interact with the nuclear pore complex, these facilitate this active transport out of the nucleus. This transport requires GTP hydrolysis. That's why it's an active transport. It requires energy in order for mRNA to be transported from nucleus to cytoplasm. Okay, RNA stability is very important when we're talking about gene expression. So although we mostly talk about translation is correlated with transcript production, so that means a higher amount of transcript will correlate with higher amount of protein as well. And that's what we measure when we are doing real-time PCR, right? So we look at the transcript levels. If higher transcript levels usually means higher protein will be translated. However, the stability of mRNA is also very important. So if mRNA is not stable, if it's getting broken down quickly, then that means the protein levels will be low because it doesn't have enough time to be translated. Whereas if the RNA is stable, if it has a longer half-life time, then that will provide enough time to be translated. So these RNA transcript levels, transcript production, as well as stability go hand in hand when we talk about gene expression. Stability of RNA is affected by these different factors. So 5' cap, secondary structure formation, because secondary RNA is very good at making secondary structure. This gives it stability, gives it a double-stranded structure instead of the single strand of RNA. Sequences in untranslated regions, so these UTRs also are very important because these provide stability, these provide the secondary structures to RNA, and the poly A tail. So all these different factors can affect RNA stability. Once in the cytoplasm, the poly A tail is actively degraded by the activities of poly A nucleases called PAN and PAP, it's called poly A proteases. So PAN is recruited to the tail by this poly A binding protein or PAP1, and they start a deadenylation procedure. This deadenylation process happens in 2 steps. First, when the PAB is bound along with this PAN to the tail, it will degrade about 20 nucleotides. Remember, the tail is about 200 to 250 nucleotide long. So the 20 nucleotides are rapidly degraded by association with PAB1 and PAN. Then PAB dissociates after degrading those 20 nucleotides, and then the slower phase of tail degradation starts where in absence of PAB1, the PAN breaks down or removes the adenines from the tail slowly. So that's how long it takes for the PAN to remove the poly A tail will also provide that will be the time the mRNA will be stable. So once the mRNA loses its poly A tail, is not very stable. So this provides stability. There are some sequences in the 3' UTR. These are called cytoplasmic polyadenylation signals. These cytoplasmic polyadenylation signals will assist PAP or poly A protease to maintain the tail. So in general, the tail will shorten over time. In general, mRNAs won't last for too long in the cell. So these in the cytoplasm, these can be stored. mRNA can be stored because you probably know that transcript can be stored, and these will be translated as soon as they get some sort of signal, physiological signal. But once these are in the cytoplasm, these mRNA degradation starts, and this mRNA degradation usually starts from the this poly A tail removal. So once the tail is removed, the remainder of the transcript is rapidly hydrolyzed and mRNA is degraded. Okay, so this is the last part of RNA processing we are going to talk about now is RNA editing. it's not always alternative splicing that can make these differences in proteins. Sometimes RNA can be directly edited as well to make different forms of proteins. So alternative splicing is a very important step. It gives a lot of diversity to provide diversity to gene functions by using the same gene sequence and produce multiple different proteins from the same genes, multiple different isoforms. RNA editing is the change in this, and it's mostly, you can call it a SNP, a single nucleotide polymorphism, a single nucleotide change that will affect the length of the protein coding sequence. So here in this example, this deamination of C that makes a U will lead to missense or nonsense modification. So this is an example of a protein called apolipoprotein B. Apolipoprotein is very important for binding to these lipids or lipases and transport those fat molecules to liver. The function of this apolipoprotein is different in liver and intestine. So these have two different isoforms for liver and intestine as well. So here this is pre-mRNA. Pre-mRNA has this codon 2153 within exon 26, which codes for CAA. All the introns are removed, are spliced out. All the exons are together here. So there is no change, no alternative splicing. However, in liver, it makes the full-length proteins because the CAA is conserved. This is required for transport of fat into the liver. So there's no editing. Translation will lead to a complete protein, full protein. Whereas in intestine, there is a different isoform. There's a site-specific deamination here, where C is converted into U. UAA is a stop codon. So this UAA will, when it's translated, it will stop here. So instead of a 4,563 amino acid protein, it will be a 2,153 amino acid protein because of this shortening here. So these are different ways this RNA can be edited, either by splicing or by direct editing. So this all about RNA processing. Next, we will talk about, once the mRNA is moved to cytoplasm, how the protein is formed. So we will talk about translation in the next lecture. And again, instead of making a new lecture on Monday, I would like to answer all of your questions up until now. If you have any questions, send those to me. I started a discussion board on Web Campus, so you can send your questions through that discussion, or you can send it directly to me by email. I will go over all your questions, and I will try to answer all of your questions, so that when we have next test, you are already prepared. Also, think about, when you're looking at this material, think about it. If you mutate any of these very important factors, what will happen to the downstream processes? Say, for example, in these slides, we have these. Let's take an example here. So what will happen if we mutate these intron-exon boundary consensus sequences? What will be the effect on transcription as well as translation? What will be the effect on gene expression? So these are the kind of questions you should be asking when you are going through these materials, because that's the kind of question I will be asking you on your test. Okay, so with that, we are done with today's lecture, and everybody be safe. Thank you.